Thank you everyone for attending. I'm going to talk a little bit about Lantern Pharma, but I'm going to spend quite a bit of time talking about how we use artificial intelligence to really develop cancer medicines that are hopefully faster to develop, de-risked in the development process, and can get to patients much more rapidly. We are a public company, so we'll make some forward-looking statements that we may or may not update. We're traded on Nasdaq, ticker LTRN. Today we have 13 lead drug programs that are powered by AI. We had 3 when we went public with Think back in June of 2020. So we've been really developing quite a few programs. We have 5 programs that are now in the clinical stage, $48 million in cash, over 95 patents. It takes us about 2.5 years to get a new molecule into a first-in-human phase 1 trial, and the average cost for programs in early stage is only about $1.5 million. These are unheard of numbers in cancer drug development. As you all know, only a few, 6 to 7% of clinical trials in oncology get through the process. It is very costly, costs billions of dollars. It's a high risk, but there are a lot of rewards if you can do it faster and cheaper. And most importantly, there's tons of data now to actually guide the development process for drugs. We use a platform that we've developed in-house called Radar. We believe that it can significantly increase our ability to get more targeted trials designed, better understanding of the mechanism of action, predict what the efficacy and accuracy is going to be of our drugs, and compress the timeline by 70% in the early phases and reduce the cost by 80% in the early phases. Again, we've launched over 10 programs in the last 2 years, so kind of an unheard of pace for a 20-plus person company. So what is our business model? One is to rescue drugs that have failed historically. We buy them and then we relaunch them into the right setting. We're doing that with one of our trials that's in phase 2. We discover and develop new drugs, including antibody drug conjugates, and we've developed a drug in less than 2.5 years, and now it's in a phase 1 trial. And then we also accelerate and de-risk programs for others. So our platform, as it's grown, has become a form of currency. So we actually have equity in other small oncology biotechs where our platform is used to de-risk or accelerate their development. We then launch into targeted clinical trials, which I'll talk about today, and then, of course, exit these programs to other big pharma or biotech companies. So in terms of our platform, I'll talk a little bit about that. Our platform today will reach about 50 to 60 billion data points by the end of the year. This is a massive increase. It was 25 billion when we started the year and less than 5 billion the year prior. Our platform has had tremendous success. It's 80 to 90% success in the types of predictions we look at. It's powered by over 8,000 data sets and over 200 advanced machine learning algorithms. If we look at how Lantern stacks, this is according to one of the analysts called Deep Pharma Intelligence. They looked at drug companies that are using AI. This is kind of the landscape. As you can see, Lantern, we're in the upper right-hand corner in terms of expertise of AI and expertise in drug discovery. We have clinical phase assets, which is usually pretty rare for a lot of AI companies today. So this is the overall framework of our AI platform. And as you can see, we get data from lots of different sources, including clinical trials, our own in vitro and in vivo studies, tons of genetic screens and panels that are available. We get information from chemical structures, from collaborations that we have, like with Johns Hopkins or Fox Chase, and also from public and private repositories. So we suck at all this data. As I said, it will be about 50 to 60 billion by the end of the year. And then we use different algorithms to ask and answer questions over and over for different types of problems. Those problems are kind of coded into modules, and I'll explain each of the modules and why they're very important. First is mechanism of action, try to understand really what is driving that molecule to work or not work. They can be very complicated, they can be very simple, but if you understand the mechanism of action clearly, it opens up the gateway to knowing how to use that drug candidate. Second is finding the right types of cancer subtypes that are most likely to respond to your molecular therapy. Third is combining it. As you know, in oncology, drug combination is absolutely essential. Putting drugs together is oftentimes the way you get above the standard of care, but which drugs and how is a really big question. It can take years to figure out. We believe we can do that in a matter of weeks or months. Next is biomarker signature creation in order to predict who are the patients most likely to respond, or what does the tumor look like from a genetic or biomarker basis that is going to respond to our drug. and then also to characterize various aspects of the molecule, like its blood-brain barrier permeability, which we also do. And then 2 new modules that we launched this year. One is for antibody-drug conjugate development, and the other is immune checkpoint inhibitor development. So these 7 modules, I would argue, are probably the most important, and they cover probably 70 to 85% of what oncology drug developers think about on a daily basis. So we believe our next goal for our platform is for this platform to become a kind of a standard. So now we're beginning to talk to a lot of big tech companies to make this available in the cloud, and so that'll be a big goal of ours in the next year or so. We've done this with Actuate Therapeutics. They're using our platform. We've published at ASCO, at AACR. We received equity in Actuate as a part of the collaboration, and we predicted their patient response with 88% accuracy, and we helped them also create a prediction panel that they're using now for patient selection and prioritization. So our roadmap, again, to reach over 50 billion, I think the next year our goal will be 100 plus to 200 billion, increase the scope of what the platform can do, and then continue to push the capabilities. ADC design is one of the most important. It can take years and years to design an ADC. We think we can smash that cost and time frame by more than 50–60%. We entered into a collaboration with the University of Bielefeld, which is part of the Magic Bullet Consortium in Germany, to do exactly that. These are some of our other collaborations: Johns Hopkins, Fox Chase, UT Health, Danish Cancer Society, and of course our biopharma companies, TTC Oncology and Actuate, both private companies where we own a small piece of them in exchange for using our platform. So here's our portfolio today. As I mentioned, our lead drug is now in phase 2. It's called LP300. The trial is called the Harmonic trial. So for those of you who know a never smoker that has lung cancer, that does happen. It's about 15% of patients. The outcome is unfortunately very, very poor. And once they relapse from initial therapy, their lifespan is fairly short. It's about 15% again of patients in the US who have non-small cell lung cancer are never smokers. In certain Asian countries, it's much higher, 30 to 35%, largely driven in those countries by EGFR mutation. We have 2 drugs that went into phase 1 trials this year. One is called LP184. The other is LP284. These are sister molecules, and they're both very unique. They have a unique form mechanism called synthetic lethality. These are very potent molecules. And then we also have an entire new subsidiary that we created earlier this year called Starlight Therapeutics. And Starlight was very interesting, and I'll take a little time out on this slide to talk about it. Starlight's sole focus will be to execute on clinical trials in neuro-oncology. When we first were developing LP184, we realized it was very potent where DNA damage response was deficient in cancers, and we've also found that it crossed the blood-brain barrier. Once we did that, we decided to go after every brain cancer in silico. So we loaded our radar platform, probably somewhere close to 2.5 billion data points, all across every possible neuro-oncology that we could find, data set. And we found that this drug worked not only in GBM, but other high-grade gliomas. It worked in a number of pediatric brain cancers, and also in brain mets, and also in ATRT. So as that portfolio of neuro-oncology indications expanded, we validated them in the lab, we validated them with partners like UT Health and Johns Hopkins, and we decided the opportunity was so significant that we should create a new subsidiary. So we'll be recruiting a management team and going after neuro-oncology indications under Starlight Therapeutics. The molecule will be STAR-001. In addition, we also have collaborations that are using our platform. So we have a very rich pipeline, which ultimately means a lot of programs that we could partner or license to pharma. So if we're not a one kind of trick pony, not just one drug. So we have multiple paths, even with these drugs, in order to get them to a stage where we can license or partner them out to pharma companies. I'll talk quickly about the programs. LP300, this is a phase 2 drug. The market potential in the US is about 1.3 billion, closer to 3 billion globally. And this is for never smokers. And so never smokers is one of the largest unaddressed cancer populations. There have been a couple of trials by big pharma where they're trying to reuse their existing approved tyrosine kinase inhibitors in never smokers. Those trials didn't work. But we believe that this drug works largely because of 2 drivers. Number one, as you saw on the last slide, the mutation frequency in certain TKI genes is very high in never smokers, and usually they don't harbor other mutations. This drug works by engaging directly with the tyrosine kinase receptors and denaturing them. That allows them to stop sucking in the TKI proteins that drive tumor growth. And the other reason it works is once the molecule is inside the cancer cell, it really restores the sensitivity to chemo. So a lot of these cancer cells, they start basically they increase glutathione and thioredoxin, and once LP300 gets in there, it resets the cellular redox cycle. And so when that happens, the cells can be attacked by chemo and be very responsive. So those are the two mechanisms that we are using. What got us excited and why we acquired this drug was because a historical company, as you saw, this was an actual trial. This is a 288 patient cohort. And as you can see, there wasn't enough increase in survival for 2-year survival or overall survival in all patients. But when you peel back overall survival and 2-year survival, which is an important endpoint, in never smokers, there was a significant increase, doubled overall survival and more than doubled the 2-year survival. At the time, though, when this drug was shelved, there was really not a lot known about why that was. There was some belief that perhaps they were more sensitive, but they didn't know the TKI profile. That was not very clear what the mechanism would be. And so as we used our engine to get more comfort with that, we relaunched the trial. The trial is a 90-patient trial. 60 patients will be given our drug, 30 patients will be given the standard of care, and we expect the enrollment to last about 15 to 20 months. Dr. Joseph Treat at Fox Chase is the lead principal investigator, and we expect to have some good data probably after the initial cohort and also after about 27 events. So we'll have some data to report out next year. And again, this is an exciting indication, and as I said, it's a high unmet need and it's growing, and no pharma company has a drug targeting this particular population. Let's talk about LP-184. This is a very exciting drug. It has nanomolar potency. It crosses the blood-brain barrier. The market potential, we think, is in excess of $10 to $12 billion. It causes double-stranded DNA breaks in cancer cells and kills off the cells as monotherapy. But the other exciting thing about this drug is that it can be used in many combinations, and these are all combinations also that we've patented. Combination with approved agents like checkpoint inhibitors, PARP inhibitors, chemotherapy, and even radiation therapy. So this has, we've got over 10 patents. We've got claims that go out into the 2041. And let's look at some of the details. So DNA damage repair deficiency. How many of you are Eagle fans in here? Some? Okay, that's okay. It's okay. Or it could be Jets fans. Actually, any pro football fan. They get loud and unruly, right? That's what cancer cells are. Solid tumor cells are exactly like that. They're loud, they're unruly, they don't really care what's going on around them, and all they want to do is multiply. And so when they multiply, they are okay with having all kinds of DNA damage repair deficiencies in their own cells. Cancer cells are. In fact, one would say that that's oftentimes why cancer cells survive after multiple rounds of therapy, because they don't really have a lot of similarity. But these unruly cells, they have a feature that can be exploited, and that is because they are deficient, about 30 to 40% of them in more advanced tumors, and about 25% across all tumors, they have a Achilles heel, and then we leverage that. We leverage the fact that they're DNA damage repair deficient, and we attack directly the DNA and cause double-stranded breaks. This occurs in pancreatic cancers, triple-negative breast cancers, bladder cancers, lung cancer. In fact, the more aggressive a cancer is, the more that we can exploit the feature of DNA damage repair deficiency. The other hallmark that happens is PTGR1 levels increase. Very important. We can measure that through mRNA, we can measure it through an enzyme, we can measure it upstream in DNA at NRF2. But when PTGR1 levels are high, that means the cancer is getting very aggressive. It stops responding to other drugs. But we use the PTGR1 to basically create an active metabolite that creates adducts directly in the tumor cells. And when that happens, this is the likely outcome. So our AI platform suggested PTGR1 was the kind of the smoke signal that we need to look for. So when PTGR1 levels were high, there was almost a direct correlation with how well our drug did. Now, on your right, no, your left, this side right here, that is in silico. So our radar platform, we ran it thousands of times, and PTGR1 is really the prime driver. You see, that was a gene signature, but really PTGR1 was the major driver. And it also predicted the higher levels of PTGR1, the more active the drug would be. And we took this data to Fox Chase, to the Pancreatic Cancer Center, and they said, well, let's try to look at the null hypothesis. Is PTGR1 really the driver? And as I mentioned before, aggressive cancers have high levels of PTGR1. That's not what makes them aggressive, but that's a signal of aggressiveness. And so pancreatic cancer has a high level of PTGR1. And so when we remove PTGR1 using CRISPR gene editing, the cell lines don't respond at all. They remain completely viable, even out to 1,000 nanomolar, which is a lot of drug for this particular drug. But in the pancreatic cancer cells that are normal, if you call cancer cells normal, they all are killed off at about 100 to 200 nanomolar. This gave us really clear, explicit confirmation that PTGR1 was really the smoking gun that we want to look for as a biomarker of interest to look at cancers. And so we're looking at PTGR1 in DNA damage repair deficiency. We have a 35-patient trial. We just dosed the first patient. Each cohort will be about 3 patients, and we'll be increasing the dose over these cohorts. We expect around patient 9 to 12 we'll get some really good response across multiple tumors, and this then drug will probably be ready to partner with a bigger pharma. So again, we think the annual market potential here is in $10+ billion just in solid tumors, and in brain and CNS cancer, several billion there. So let's look at what some of the data has been. Again, pancreatic cancer on one side. These are unfortunately actual mouse models, mouse that were injected with pancreatic cancer. And as you can see in A, the tumors become quite aggressive. But when we dose them with the LP-184 in both cases of 2 different types of pancreatic cancers, they almost diminish. We also looked at triple-negative breast cancer, again, an aggressive cancer, and we looked at triple-negative. And again, these were triple-negative breast cancer mouse models. And these mice, as you can see, they stop responding to PARP inhibitors, which is what's given today for many of these TNBCs. In the gray, you can see that the tumor volume continues to grow when dosed with PARP. And when dosed with our drug in the red, you can see these tumors almost across the board just vaporized. So we saw that there is something really unique. We got orphan drug designation in pancreatic and multiple other indications. Again, we can combine this drug with other existing standard of care agents, and we think it's going to be a blockbuster franchise. We looked at this, again, in DNA damage response deficient cancers, both NERD and HERD cancers. We see large-scale sensitivity. Again, the nanomolar sensitivity is in the double-digit range, and it shows exquisite potency in across lots of solid tumors. And also, we can combine this drug with spironolactone to increase its efficacy, and we can also make it work with radiation therapy. So this drug, we believe, has a lot of potential across the entire range of solid tumors, both as monotherapy and combination therapy, and we think would be an excellent asset to partner with large pharma. LP-284, this is a sister molecule. This molecule didn't exist 3 years ago. In fact, it was about later in Q4 that we first started brainstorming this molecule, because we saw how potent LP-184 was, but it didn't work in blood cancers. This molecule works only in blood cancers, and it doesn't work in solid tumors. So the pharmacophore is very similar, but again, this went from whiteboard to now a first-in-human trial in less than 3 years with a cost of $2.7 million. That includes GMP manufacturing of the drug. We think the market potential here is about $3.5 to $4 billion globally. Let me share some of the data. Non-Hodgkin's B-cell lymphomas were the 7th leading cause of cancer in the US, even higher in Europe. And mantle cell and high-grade B-cell lymphomas, we applied for orphan designation. We have it already in mantle cell. And as you can see, mantle cell in the red, across a wide range of mantle cell lymphomas, this drug was super potent. And so now it's in a trial. We've already initiated the trial. We expect the first patient to be dosed this quarter. And we publish at ASH, ASCO, et cetera. Here's the chart that got me very excited about it. And if you look at the red and blue bars, Those are big billion-dollar drugs, bortezomib and ibrutinib. I think bortezomib might be $800 million now in the US, but these are very bad drugs. They're very toxic, but they help mantle cell cancer patients. But eventually, those mantle cell and other high-grade B-cell lymphomas become resistant. It's not a matter of if, it's when. When they become resistant, the tumors grow. And as you can see in the red and yellow bars, the tumors grow quite aggressively. And when we dose them with our drug, the tumors virtually disappeared. And so these are, again, tumors that are resistant to standard of care. And so that got me very excited because now we can go right after ibrutinib and bortezomib in the second or relapse setting. And that is really a great place to be initially for B-cell lymphomas. So we're very excited by it. And as you can see, our dosing, if you look at the arrows at the bottom, our dosing was far fewer than ibrutinib. And because it's doesn't have the toxic side effects that bortezomib does, we think it has a better therapeutic window as well. So again, some data we just presented a few months ago at SOHO, and as you can see, LP-284 in the red and yellow, plus rituximab, which is a standard of care drug in B-cell lymphomas, nearly wipes out these tumors even a month after dosing. So again, there's no standard of treatment for high-grade B-cell lymphomas. So this is a wide open opportunity. We think the drug works really well. We think we'll prove that out in the early phase trials and partner the asset. As I mentioned, I was very excited about Starlight. I'll take a couple of minutes because this is a major value driver for investors and for the company. As I mentioned, we're constantly spinning up new ideas for indications and for drugs. Starlight was focused entirely on CNS cancers. And there's a lot of patients globally and many neuro-oncology indications that don't have any standard of care or any treatment. 5-year survival rates across neuro-oncology indications are miserable. And our drug seems to work in many of them. So GBM, high-grade gliomas, brain mets that arise from breast cancer, from small cell lung cancer, and melanoma, and also in pediatric brain cancers where there's really no drug. And so phase 1A work will all be done by Lantern, and then we're going to spin this out. This will be a phase 2 company out of the gate next year, and we'll be in trials in a couple of the major centers. We're presenting in a few weeks at the Society of Neuro-Oncology with Johns Hopkins. This drug has excellent blood-brain barrier permeability, excellent bioavailability, much better than other brain cancer drug, standard of care drug called temozolomide. And it has, again, nanomolar potency, so we can have multiple shots on goal. So as I mentioned, we're pretty excited. We continue to spin up new ideas, both indications and new molecules. We have over 95 patents. We've got a lot of orphan drug designations very early because we have characterized these molecules and taken them to a lot of very interesting models to show their development potential. We also have pediatric rare disease designation, which if we get that drug approved in ATRT or other pediatric brain cancers, we can sell that voucher. Vouchers go for about 100 to 120 million. Lots of posters and publications. There was one that just came out in Clinical Cancer Research a couple weeks ago in GBM. And so we're at all the major cancer conferences and this helps us because we always publish with thought leaders and helps us de-risk the development, downstream development. $48 million in cash. Our quarterly burn is somewhere in the range of $3.5 to $4.5 million a quarter. I've got 10.8 million shares and 12 million on a fully diluted basis. We have cash for the next several years and enough to really reach the milestones that we want so that we can partner out the assets for several hundred million or more. And so that is our model. Take a molecule, take an indication, spend 5, 10, 12 million, get it to meaningful clinical data, and then sell it for 10 to 50 times that amount. We think we can do that over and over and over and over in cancer. And then more importantly, as the platform grows, we think the whole platform can be spun out as its own standalone company for all cancer companies. Because it's expensive to make a platform and it's really time consuming to make a good one. And we've seen through our own development that our platform is becoming more and more powerful, and it solves a lot of the fundamental problems in cancer development. Now, every disease is different, but we've really curated for a cancer, and it's going to continue growing. The great thing about our platform also is the platform actually now is growing by itself. So the platform wakes up, it finds what data is interesting to us as we feed it into the data table, and it asks us if it wants to pull down data. We're really interested in certain single-cell genomic data in pancreatic cancers and certain pediatric tumors. It'll find studies and it'll scrape in the data for us, and then we can accept it or not accept it. The algorithms, there are 200 algorithms now that power it, and the algorithms are now also doing what AI should do, which is write themselves. And so now that we've curated all these algorithms, now the algorithms are also learning and writing themselves. This is a team. David Margrave, our CFO, is here if you have any questions for him, especially intricate questions about spend per month per program. He likes to have some charts for that. Our Chief Scientific Officer, Kishore Bhatia, was formerly at the NIH. Peter Carr is our Principal Software Architect, who's at the Broad Institute and was the lead programmer for Gene Pattern. And Reggie Avasuda is our Head of Clinical Development, and he's been at Pfizer, Tessera, GSK, a lot of great companies. So we've got a great team that's kind of mission built for exactly doing what we're doing, which is using AI to transform the cost and the timeline of cancer drug development. Any questions? Yes.
So, in what phase did you start spinning out pharma parts?
I think phase 2 is the best phase.
Okay.
Yeah, because the way I look at it, and I was an investment banker in life sciences, the typical rule of thumb for oncology assets is that if you have a phase 3 asset that is, that you're waiting for PDUFA approval and you have good data, they'll typically pay about 4 to 6x peak sales. You can thumb wrestle over what you think peak sales. You might say it's $1 billion. They'll say it's 500 million. You say it's 2 billion. They'll say it's 750, whatever it is. But it's about 4 to 6x peak sales after phase 3. At phase 2, it's a 50% discount. Phase 1, it's about a 90% discount. That's the rule. There are, of course, dots on both sides of those, but that's kind of the rule of thumb I think about. And so phase 2 is great because we have money to do phase 2. So if we can do targeted phase 2s and show really good efficacy, which we think we can with these molecules, I think there'll be a line of pharmas for these indications.
Thank you, Panna. I appreciate you joining us today. Thank you, everyone, for your attendance.